Hello! In this video, we're going to learn about cell division. Before we begin, you might be wondering why we need to study cell division. The answer is simple. Cell division happens in our bodies all the time. It's what allows us to grow, heal wounds, replace old cells, and reproduce. When this process doesn't work properly, it can lead to problems such as cancer and other diseases. For students in medical and health related fields, this is one of the most important foundations we learn because many topics you'll study later are connected to it. And of course, when someone asks you how a wound heals, how a baby develops, or why cancer happens, you'll actually know what's going on and be able to explain it. Cell division is a broad topic, so in this lesson, we'll focus on understanding it as a whole first. Then we'll break it down step by step into its three main parts: mitosis, meiosis, and cytokinesis. Since there's a lot to cover, we'll go through it step by step, and each part will be discussed in separate videos as part of this lesson series. Cell division is the process by which cells produce new cells. This process is important because it allows organisms to grow, repair damaged tissues, and produce reproductive cells. There are two main types of cell division: mitosis and meiosis. The first is mitosis. Mitosis produces daughter cells that are genetically identical to the parent cell and is responsible for growth and tissue repair. For example, when you get a small cut on your skin, mitosis helps replace damaged cells with new ones. The second type is meiosis. Meiosis produces gametes or sex cells such as sperm and egg cells. These cells contain half the number of chromosomes found in the body cells. Meiosis also contributes to genetic variation, which creates unique combinations of traits and offspring. For example, meiosis is one of the reasons why siblings can look different from one another. Now, before a cell divides, it goes through a series of stages known as the cell cycle: G1 as one phase, G2, and G0. The longest stage of the cell cycle is called interphase, which occupies most of cell's life. During G1 or gap 1, the cell grows and performs its normal functions. Think of this as gathering materials before starting a school project. Next is the S phase. During this stage, DNA replication occurs, meaning the cell makes a complete copy of its DNA. This is similar to photocopying important documents before sharing them. After that comes G2 or gap 2. The cell continues growing and prepares for division. Imagine checking your project one last time before submitting it. Some cells enter a stage called G0 where they remain metabolically active but doesn't divide. For example, it's like a parked car with the engine still running. It's still active, but it's not going anywhere. Throughout the cell cycle, checkpoints monitor the cell and ensures that conditions are suitable before moving to the next stage. You can think of checkpoints as quality control stations in a factory. Now, let's focus on mitosis. Mitosis consists of four major phases: prophase, metaphase, anaphase, and telophase, or the PMAT. First is prophase. During prophase, chromatin condenses into visible chromosomes and spindle fibers begin to form. Think of it as organizing loose papers to folder before distributing. Next is metaphase. During metaphase, chromosomes line up along the center of the cell. Imagine students lining up in a straight row before entering a classroom. Then comes anaphase. During anaphase, sister chromatids separate and move toward opposite poles of the cell. Think of identical twins holding hands and then walking to opposite sides. Last, we have telophase. During telophase, new nuclear membranes form around each set of chromosomes, creating two daughter nuclei. This is similar to dividing one large room into two separate rooms. After mitosis comes cytokinesis. Cytokinesis is the division of the cytoplasm, resulting in two separate daughter cells. Think of cutting a cake into two equal pieces so that each person receives a slice. Now let's move on to meiosis. Unlike mitosis, meiosis involves two rounds of division, meiosis 1 and meiosis 2. During meiosis 1, homologous chromosomes separate, reducing the chromosome number by half. In humans, this reduces the chromosomes number from 46 to 23. Example, splitting paired items. Unique event called crossing over occurs during phase 1. During crossing over, homologous chromosomes exchange segments of DNA, creating new genetic combinations. Think of two friends exchanging stickers to have unique collections. This process increases genetic variation. During meiosis 2, sister chromatids separate, similar to what happens during mitosis. Think of separating two photocopies into four individual copies. The result is four haploid daughter cells, each containing half the original chromosome's number. Now, let's discuss what happens when the cell cycle is not properly regulated. Normally, cells divide only when necessary. However, defects in cell cycle regulation can cause cells to divide uncontrollably, which may lead to cancer. You can think of this like a car with faulty brakes that cannot stop when needed. Cancer cells often shows abnormalities such as irregular nuclei, prominent nucleoli, abnormal chromatin patterns, and uncontrolled growth. Next, let's learn about the important structures involved in cell division. Chromatin is the loosely packed form of DNA found during interphase. Think of it like a long piece of yarn that hasn't been rolled up. A chromosome is condensed DNA that becomes visible during cell division. It's like rolling that yarn into a neat ball. A chromatid is one copy of duplicated chromosomes. It's like one copy of a document. Sister chromatids are two identical copies joined together. They're like two identical popsicle sticks attached together in one pack. The centromere is the region that holds sister chromatids together. It's like the staple connecting two sheets of paper. The kinetochore is a protein structure where spindle fibers attach. It's like a hook used to pull a box. Spindle fibers act like ropes that pull chromosomes apart. Centrosomes and centrioles organize the spindle fibers. They're like construction supervisors directing workers. Last, telomeres are protective structures found at the ends of chromosomes. Think of telomeres like the plastic tips at the ends of shoelaces that prevent them from fraying. Now, let's take a closer look at chromosomes and chromatin. Chromosomes are long DNA molecules wrapped around proteins called histones. Histones help package and organize DNA efficiently inside the nucleus. Most human body cells contain 46 chromosomes arranged into 23 pairs. Before cell division, DNA exists primarily as chromatin, which is loosely packed and accessible for normal cellular activities. As cell division begins, chromatin condenses into chromosomes so genetic materials can be distributed accurately. Think of organizing scattered papers into folders before handing them out. Next, let's understand how healthy cells decide when to divide. Cells divide only when needed and when they receive appropriate internal and external signals. This coordination is essential for proper growth, development, tissue maintenance, and repair. Not all cells divide at the same rate. Some cells remain in G0 and may never divide again, while others can re enter the cell cycle when needed. Cell cycle checkpoints monitor several important conditions, including DNA integrity, cell size, nutrient availability, and signals from neighboring cells. Think of these checkpoints as inspectors ensuring that every step is completed correctly before moving forward. Now, let's summarize the major steps of the eukaryotic cell cycle. First, the cell grows. Next, it replicates its DNA. Then, duplicated chromosomes are distributed. Finally, the cell divides. A simple way to remember these steps is growth, replication, distribution, and division. Before we move on, here are a few additional notes about the eukaryotic cell cycle. A typical eukaryotic cell cycle takes about 24 hours to complete. About 95% of that time is spent in interphase, while mitosis and cytokinesis together take only about 1 hour. This shows that cells spend most of their time preparing for division rather than actually dividing. Most of the cell cycle is spent in interphase, while mitosis and cytokinesis occur during a much shorter period. Like students working on a project, most of the time is spent researching and preparing, which is like interface. Only a short amount of time is spent presenting the project, which is like mitosis and cytokinesis. And that concludes our lesson on cell division. To summarize, cell division enables growth, tissue repair, and reproduction. Mitosis produces genetically identical daughter cells for growth and repair, while meiosis produces haploid gametes and contributes to genetic variation. Through checkpoints and careful regulation, cells ensures that division occurs accurately and efficiently. And that is the end. Thank you for listening and see you in the next one.